Hey guys, happy Monday. Hope you guys had a good weekend. If you missed live class, this video is going to tell you what we're doing Monday. We are building on what we've been learning about genes. How do we inherit them? What traits do they give us? What happens if you inherit a different trait from mom and dad? What might happen? So today I'm going to introduce you to one of my favorite tools a geneticist can use, and that is called a Punnett square. It's going to make a really simple question out of more complicated questions. So here's a little bit of the complicated question. Then later, I'm going to teach you a really simple tool to answer a question like this. So in this situation, brown eyes, we could use the big B allele. Are dominant to blue, we'll use the little b. Remember, big B, capital letters are dominant, lowercase letters are recessive. So what gene combinations, and when I mean that, by that I mean big B, little b, or little b, could the baby here in this model inherit from his mom and his dad? So I want you to tell me what combinations could baby inherit. You could definitely do that. It won't take you that much time to figure that out. Number two is a little bit trickier. You could probably do it without a Punnett square, but it's easier if you use one. But number two says, what is the percent chance that baby will inherit blue eyes? That's a little bit harder. Can you tell me? Give it a shot. Pause and definitely answer number one, and then we'll keep going. So after our scientist of the day today, I'm going to teach you how to use Punnett squares. In our breaker room, we're going to do our first Punnett square. You guys will do one together, or at least they did. If you missed live class, I'll do it with you real fast. And your independent work is up to you to do some Punnett squares together, okay? There actually is no exit ticket today. We will continue practicing Punnett squares on Tuesday. This is Cliff Capono. You might be looking at his birth date to find out how old he is. He's a professional surfer, he's a journalist, and he's a chemist. He has a PhD in chemistry, and he was born in Hawaii, and he lives in Hawaii today. He's a part of this big project called Mega Lab, and they study coral reefs. And they try to learn how healthy it is. They look at the chemistry of the ocean, how it's changing. And he is big on sharing his research. He has like websites. He does digital media. You can follow him. If I move my face out of the way, you can see you can follow him at his website here. I saw him on a commercial, which is really cool. He produces award-winning films like about indigenous activism, ocean conservation, global food security, virtual reality. He's just in all kinds of stuff. There's a couple cool pictures of him. There's a picture of him surfing. He's legitimately a professional surfer. And this is him on the right here. He's doing a podcast. So he tries to get himself out there. He tries to make himself available for you to learn from. About the ocean, about the chemistry of the ocean, about the living things in the ocean, about a lot of other things apparently as well. So there's Dr. Capono. Okay, so today a couple of new vocabulary words. Genotype are the gene combinations, and then is. You can only have one genotype. You have a genotype for specific alleles. So the gene combination an individual has. For fur color, Snowball's genotype is little f, little f. Or you can say two little f's, or different ways to say that. Technically, it's it's two recessive alleles, another thing you could say. Homozygous recessive. I'm not going to make you learn that term. Smokey's genotype is big F, little f. These are the types of genes you can have. That seems pretty easy, right? The types of genes. Genotype, okay? So what about phenotype? The phenotype is the physical trait an individual has. So remember that pH for pheno is like the pH in physical. That's how I remember it. Snowball's phenotype is white fur. So if you were asked, like, what's its genotype? Oh, the genotype, the genes it has are two little f's, or two recessive alleles, or little f, little f. But what's the phenotype? What's the physical trait? White fur. See how we're asking slightly different questions there. What genes do you have? You can't see genes. Genes are like inside your cells. You need to be able to like do tests to find out what genes somebody has. Or you can do it by some clever crosses with the mice. You can know. Smokey's phenotype is gray fur. You can look at Smokey to determine the phenotype. You can tell physical traits by looking at them, but genotypes require more. Okay, so we're going to practice those today. Okay, I got 4 slides to teach you how to use a Punnett square. If we wanted to know the chances the baby will have blue eyes, we could use a Punnett square. And a Punnett square looks like this. Looks like a window. So these are actually the 4 slides. Okay, so during sexual reproduction, a sperm and an egg come together and they make a cell. And that cell becomes an offspring. If the sperm and the egg had all the genes and the chromosomes of both parents, then the baby would have twice as many chromosomes, twice as many genes as the parents. And then going on and on into the future, you realize that you have like twice as many, twice as many, twice as many. Within 5 generations, you have 10 times as many chromosomes. That'd be a crazy mess. So that's not what happens. Instead, each sperm and egg only have one version of each gene, of each chromosome. Remember, the chromosomes are pieces of the gene. So, sorry, that's not true. The genes are pieces of the chromosomes. So the big molecules are the chromosomes. You get 23 from mom and 23 from dad. And that big molecule is like a book. And in that book are a whole bunch of little genes. Okay, so these are mom's eggs. If mom has big B, little b, then half of her eggs have a big B and half of her eggs have a little b. But no eggs have both. Dad doesn't have eggs, he has sperm. Well, actually, since he has only little b alleles, all of his sperm have one little b allele. And remember, when the sperm and the egg form together, they make a baby. So you can see if these two babies were made, that baby would have a big B and a little b. If you have these two, that baby would have a big B and a little b, or sorry, 2 little b's. Okay, so this is how a Punnett square works. We need to know the genes that mom and dad have. We know those, okay. Then we put on the outside of the square mom and dad's genes. So we can put dad's genes here. Remember, these are like the 2 possible sperm that dad can produce. Sperm with little b's or sperm with little b's. These are the 2 possible eggs mom can produce. Big b's or little b's. And then these are the boxes are the possible ways they could combine. So to show how they combine, we move the big B down, or sorry, the little b from dad down. And we move the big B and the little b for mom over. Look at those arrows. This is just coming from the top, coming from the side for both. So anytime you look at a blank box, you can look up to see the little b and look left to see the big B. And then that's what you write in here. This is just like how a multiplication table works. 2 times 2 is 4. 3 times 2 is 6. Here, little b times big B equals big B, little b. Works the same way. Then each of these boxes represents something. Each of these boxes represents 25% chance of an offspring having that allele, that genotype, and also then that trait. So then the question we go back to, what are the chances that this mom and this dad have a baby with blue eyes? Well, these are the 4 quarters, the 4 25% chances. So which of these would have blue eyes? These 2. Remember, you need 2 little b's to have blue eyes. If you have a big B, you have brown eyes like mom. The big B is dominant. It overpowers the little b. So what percent is this? It's 50%. Or a 2 of 4 chance, or half a chance that this mom and this dad would have a baby with blue eyes. There you go. That's how you use a Punnett square. So in breakout rooms, people are doing this, and so let's do this together right now, since you missed live class. So setting up a Punnett square, and then we're going to answer these questions. A tall plant, big T, is crossed with a short plant, little t, little t. Okay, so I'm going to put big T across the top. And I'm going to put little t off to the side. Does it matter? Could I put the little t's up here and the big T's here? Of course. It doesn't matter. Remember, it's like multiplication. 3 times 2 is 6. 2 times 3 is 6. Oh, it doesn't matter which one comes first. Okay, now I got to fill this in. I look up, I see a big T. I look left, I see a little t. I look up, I see a big, I look up, I see a big T. I look left, I see a little t. Up here, I see up, I see big T. Over, I see little t. Okay, big T, little t. Up, I see big T. Over, I see little t. Oh, they're all the same. What do the letters, oh, you know what? I didn't add arrows in, so I'm going to do that now. What do these letters represent? Those there letters. Let's do it pink. I like pink. What do those letters represent? These letters represent the alleles of the parents. I have the parent with big T, and I split the alleles up, big T. What does each of these boxes represent? These boxes each represent a 25% chance that an offspring will have this genotype and the given phenotype that's associated with it. So each of these boxes is 25%. What does a Punnett square help us do? You can use Punnett squares to predict the possible outcomes of a genetic cross, we could say, or the possible genotypes and phenotypes of offspring of two given adults, something like that. Okay, and so for this specific question, what percent of the offspring will have short plants? Will be short plants, yeah. I'll just make it short. What percent is it? It's 0%, because none of them are short. Because all of them have a big T. So they're all tall. So there you go. Okay, so we're going to practice this. You're going to probably need to go back up and revisit slides 7, 8, 9, and 10. That's okay. This is one of those things that you do the first one, you're like, what am I doing? This is hard. And you do the second one, you're like, oh, I kind of get it, but it's still in the third. And then by the time you do like your 30th one, you're like, this is super duper easy. And each one can only take a few seconds. So 30 won't take you very long. And so we'll spend a couple days getting really good at these, okay? So there's 4 here. This one's about Snowball and Ash. This one's about Cloud and Smokey. This is about a Pip and Squeak. This is about some Freckles. That's it. I want you to do these 4. I want you to come Tuesday, ready to do some more, ready to talk about these. And that's it. Thank you, guys.